cytosine pairs with thymine in a double hydrogen bond, and guanine and cytosine pair in a triple hydrogen bond. And so the DNA replication occurs during during cell replication in interphase before meiosis, mitosis, or binary fission.
Okay, so starting the replication, if you would imagine that these two strands are in this double helix shape, then an enzyme called helicase will come through and unzip the two strands by breaking the hydrogen bonds in between the nitrogenous bases. Then, there it is, DNA polymerase III would come and follow it, and it would add complementary nucleotides that are found floating around in the nucleus.
It would add them to the leading strand here, and it would add these bases.
And it has to, and it follows the helicase because it has to go in a 5' to 3' order, and that's the way it goes on the leading strand.
And for the lagging strand, it's a little more difficult because the lagging strand's parental DNA goes from a 3' to 5', and because they have to be complementary, it has to go from 5' to 3' in this direction. So the DNA polymerase forms DNA this way, in opposite direction as the helicase. But before it can do that, we need RNA primase to lay down an RNA primer, which is basically a foundation before the DNA starts to form. So the green icing is the RNA, and as you can see here, there's a pink dot instead of a green one because uracil replaces thymine. So after the RNA primer is laid down, the DNA polymerase forms DNA and adds nucleotides. And so as it keeps doing that as helicase keeps splitting, so there's more empty space here. So RNA primase has to keep adding more RNA, so DNA polymerase can keep adding more DNA. And this forms different parts and different segments, and these segments are called Okazaki fragments that are separated here. Then the DNA polymerase I comes and replaces the RNA with DNA, and then after that, DNA ligase comes and connects the Okazaki fragments.
And the result is, this process is semi-conservative because it uses the strands from the original DNA, but it also adds new strands of DNA from the DNA polymerase III.
And even though they're occurring on different strands, DNA replication, both of these happen simultaneously, and that helps DNA replicate really fast so that it can do what it needs to do and not waste time splitting. The end.
Thank you.